Good morning, everybody. Thank you very much for joining us today's webinar on chalk stream species at risk. Very shortly, I'll be handing over to Alison Matthews, chalk stream restoration program manager at the Rivers Trust, who will be chairing this session. Just very quickly from myself, my name's Jack. I'm from the Rivers Trust, communications assistant for media and events. I'm just going to run through a couple of very quick housekeeping points. If you want captions, you can do that just by looking at the little bar at the bottom of the screen. Click more, and you should see a captions button there, so you can get captions up. We will also have a live Q&A at the end. So if you have any questions, please do have those ready. You can pop those in the chat if you want to, and then we can go for a Q&A right at the end. We are also recording this session, so just be aware of that. And I will hand over to Alison, who will get us started. Thank you.
Good morning, everybody. Really pleased to welcome you to this next in our webinar series. So I think this is our 7th one now. So we really welcome contributions from around the Chalk Stream network for our webinars. And today is no exception. We've got presentations from around our chalk streams. We're going to start off with Curtis talking about the Atlantic Salmon Trust project to save the chalk stream salmon from extinction. Then we're going to move on to Debbie at Nature Safe to talk to us about something I don't know much about actually, which is cryo-biobanking and the work they've been doing with the Chilterns team to protect crayfish. Sarah Teasel is going to take us on to talk about a really interesting project about the crystal moss animal. Again, another thing that I've never heard much about, so I'm really looking forward to hearing Sarah. And then we're going to finish off with Sarah Perry, who's going to talk to us about a project that Herts and Middlesex Wildlife Trust have been doing to protect the whole river habitat. So we're going to start off looking at sort of species at risk, individual species at risk, and finish off looking at projects to protect the habitat that the species need. So we've got a really sort of varied and balanced programme across from north to south and east to west. And I think probably without further ado, I'm going to hand over to Curtis to start us off. And as Jack says, do please remember to put anything you sort of any chat about where you're from, or say hi if you want to in the chat. But if you've got any questions for the Q&A session, if you can put them in the Q&A, we'll come to those at the end, and our panelists will pick them up as they go if they can as well. So I'm going to hand over to Curt. So there you go, Curt.
Lovely stuff. Thank you, Alison. I will just share my screen and then get going. Right. Sorry, 1 second. Just need to make sure I've got everything up that I need. Okay, right. Can everybody hear me all right?
I can hear you, Curt. Can't see your slides yet.
Oh, okay. Sorry, 1 second. Is that better? I didn't click share.
Yeah, if you put it on slideshow mode, we're good to go, I think.
Lovely stuff.
Fabulous. Thanks,
okay,
Curt.
We're in. Okay. So I'm going to talk a little bit today about England's vanishing wild salmon. So as this lovely little salmon par here is showing us, this is the distribution of chalk rivers in England, from Dorset down in the southwest-ish, across Wiltshire, Hampshire, up the Thames Valley, through Norfolk, and then to the east coast of Yorkshire. I'm not going to go into too much detail about chalk rivers right now, because I'm sure, as you are all fellow chalk stream nerds just like myself, you tell people all the time that they're the rarest freshwater habitat, or one of the rarest freshwater habitats in the world. It's my go-to fact when I tell people about these rivers. But salmon are actually confined to quite a small area of these chalk rivers. They can actually just be found on 7 of these rivers, from the Froome and the Piddle and the Dorset Stour, over to the Avon, and then the Test, the Itchen, and the Meon a little bit further east in the Hampshire Basin that feeds out into the Solent near Southampton. So quite a rare group of fish found in a very specific area of England. Now, I'll talk a little bit about the project. I'll introduce it. So yeah, Project White Hart is this growing coalition of local and national organisations shared by this mission and this vision to save chalk stream salmon So it really started in 2025, where the kind of original mission goal is to halt the imminent extinction. And then looking forward with this big vision by 2050, we want to see thriving populations of chalk stream salmon. So my name's Curt and I'm the project coordinator for this project. And as you can see, it's fairly new. It's in its infancies. So things are just getting going. But I'm going to go on to talk a little bit about why chalk stream salmon are so special and what some of the goals are for the project going forward as we look to that long-term plan to 2050. So the project is focused around these three rivers in the Hampshire Basin that I just talked about, the Test, Itchen and the Meon. They form a meta-population between the three rivers, which I'll talk a little bit more about in a minute. So yeah, the project is really focused on these three rivers. And why are we looking at these salmon? Well, the situation is critical. They really are on a path to extinction. Salmon have been classified as endangered, and populations are generally declining across the board. And it's a real shame because they're considered an indicator species. And if salmon are doing well in a system, then it's highly likely that the river system itself are doing well. And this biodiversity emergency is really highlighted by this fact here about on the River Itchen in 2022, just 133 adult salmon were counted returning to spawn. So we're really in this kind of cultural, this critical phase for salmon on these chalk rivers. And it, as well as being this biodiversity emergency, it's also a cultural tragedy. Salmon have played a real big part in the culture of these chalk rivers for a long time, and people have come from around the world to fish them. Local economies built around it and culture built around it too. So a loss of it would not only be this biodiversity situation, but also a cultural one at the same time. Now, digging into this decline of salmon a little bit more, there's this really fantastic data set that goes back to 1990. This is based on fish counters that were installed by the Environment Agency at the bottom of the Test and Itchen. And they've been managed since then by the Environment Agency, producing yearly reports with stock estimates and conservation limits being calculated. And it's an incredible data set that takes up a lot of time and knowledge and resource just to upkeep these fish counters. So we're quite lucky to have this data set on these rivers. As you can see, the test is in blue and the Itchen is this kind of yellowy brown. The test has always had a slightly higher population of returning adults. Funnily enough, the rivers aren't massively different in size. The test is a bit bigger and it does have more available habitat, but the difference is fairly stark. So there is likely some stuff going on in the Itchen that isn't happening on the test that is leading to this particularly lower population. And as you look at this graph, you can see that 1 year in particular has quite a stark difference. And it's this 2020 year, you see this huge rebound in returning salmon, particularly on the test. Almost 3,000 adult fish were recorded coming back. And if we cast our minds back to the year, it was, of course, the 2020 lockdowns due to COVID. So really, this is a visualization of this reduction in human pressures on salmon, particularly at sea. The sea phase, the marine phase of salmon, they're obviously subject to fishing and bycatch. And there was a real reduction in this during this COVID year. So it really highlights how a reduction in human pressures can instantly have quite a stark impact on these populations. But unfortunately, this 1 year of rebound did not lead to long-term recovery. And actually in 2022, this was this year that I mentioned a second ago, we saw some of the lowest numbers of returning salmon to the Itchen, just 133. So even with 1 year of relief, we didn't see any kind of rebound. We've actually seen some of the lowest numbers. This graph shows this data in a slightly different way. This is the compliance limit, the conservation limit, which is calculated by the Environment Agency, and it uses an egg deposition estimate based on the amount of returning adults and sex ratios and sizes of those adults. Quite a complicated calculation from what I understand. And any value under 100% indicates that the population is depleted and that it has low resilience to continued pressure and it needs urgent intervention to restore stability. So going back to this 2022 figure of just 133 salmon on the Itchen represents just 20% of this conservation limit. So it really is a critical point. But why should we care about these chalk stream salmon in particular? Why are they special? So there'd been some research done on this before, but a recent paper came out in 2026, which studied the genetics of Atlantic salmon populations. And the results of that proposes that Atlantic chalk stream salmon should be classified as its own unique subspecies, um, with the suggested name of Salmo calcarensis, if my Latin is serving me all right there. Um, and I really recommend taking a look at this article. I can circulate it after, as well as Wildfish put out a really good article which breaks down the paper into a bit more of an easily digestible summary for those that are not geneticists like myself. So I will circulate that too. And this is one of the outputs from that study. You can see here that there are genetically distinct groups of salmon. Here you have the English chalkstream salmon, and these are the French chalkstream salmon, which are on 3 of the French rivers. And then over here you have the more normal English salmon rivers, as we would call them. These ones in red being Devon and Cornwall, over to the Irish Sea rivers. So geographically, these rivers are really close, but you can see that there's a genetic distinction between them. And some rivers are actually shown to be almost genetically identical. So the Tesme and Anichin here come off on this own little branch. And this is really what I talked about earlier about the project focusing on these 3 rivers. They form this meta-population with some straying of individuals between the 3 rivers. And the other thing that makes them quite unique, as well as some ecology, which I don't quite have time to get into today, but these chalk rivers and the salmon populations within them, they persisted through peak glacial max during times when the Welsh and the Irish, the Northern English and the Scottish rivers were covered with glaciers. So when I talk to some of my counterparts at Atlantic Salmon Trust and they tell me about some of the real big salmon rivers of Scotland, I gently remind them about the fact that chalk stream salmon are actually some of the UK's oldest and most ancient populations. So the high genetic uniqueness of chalk stream salmon prompts the discussion to recognise them as a distinct subspecies. This is important to drive legislation in the UK and guide conservation efforts. And the study found that 6% of unique genetics would be lost if these populations disappeared. So we really can't replace these unique fish. So going back to the project, what are we going to do to save these salmon? It's quite a bold claim, of course, but we've set the project up in a coalition. It's locally led by the Test and Itchin Catchment Partnership, with the joint chairs being Wessex Rivers Trust, Hampshire Night Wildlife Trust, and Atlantic Salmon Trust. And then we're supported by this wider group. We're funded by Southern Water until 2030, which covers the catchment audit and the restoration strategy, along with a salmon recovery fund, which we have for some small and more immediate wins and projects on the rivers. And yeah, we've been supported really well by these groups. GWCT, Test and Itchin Association, Anglin Trust, Environment Agency have all been really good to us in establishing this project, sharing data and the like. And we couldn't have done it without them at this point. So what are the phases of the project? What's the plan? So initially, we had a stage of ongoing stakeholder engagement. That was the engaging with communities and NGOs, the regulators and policymakers to ensure that a project like this could actually be established and actually work in this context. And then stage 2, really kind of where I came in, I started in the role November last year, along with my colleague, Maya Moreni, who's our data and GIS specialist. We're brought in to do the catchment audit, where we're bringing together all the information that's available on these catchments to produce an ecological health check with a salmon lens, I like to say, to define what good looks like on these rivers. And then going to the next phase, it will be launching a program of works based on the restoration plan to hopefully secure a thriving future for salmon on these rivers. So just delving a little bit into what catchment audit means. It's quite a vague term, but really it's about bringing together existing data that is on the catchments. Quite lucky on the Test and Itchin, there's lots of work that's already been done on these rivers by the Environment Agency and local groups, community groups. So bringing that all together and assessing it with that salmon lens. The continued stakeholder engagement. and also capturing some of the lived experience of people who have lived and worked on these rivers for a long time, which often isn't captured in these data sets. So really thinking about how we can bring them people onto the project and taking that data that's in their brain and incorporating it into the audit. We're also looking to do some modeling using the skills at AST, some forward population modeling, sorry, as well as potentially finding data gaps within the catchment and rolling out additional monitoring schemes. Now, I'm just going to touch a little bit on some of the themes of the outcomes of the catchment audit. I've basically picked these because I have some nice pictures to go along with them. This is not the exhaustive list, and there will be a lot more detail than just some of these pretty pictures at the end of the catchment audit. But really, some of the things that we'll be looking at and the impacts on salmon will include the impacts of pollution and water quality, the impacts of low flows and over abstraction, looking at rates of predation, particularly when thinking about compounding effects of barriers and man-made structures. And yeah, the urban infrastructure and barriers such as road runoff, artificial channels, and considering the highly urbanized environments of Southampton and Winchester, for example. Early assessments, we found potentially up to 200 barriers on the Itchen itself. Some of those being minor, but others being what I like to call big bad barriers, which are definitely going to be having a severe impact on salmon. Considering things like poaching and illegal fishing, as well as invasive species, looking at egg survival, and particularly high levels of siltation within the rivers. There's already been some really amazing work done by the academics at Southampton Uni and PhD students looking at quite a stark level of sedimentation affecting salmon egg survival on these rivers. So hopefully next year I can touch on this in a little more detail, the impacts and each of these themes. I'm just going to talk a little bit about finally about what we are kind of trying to view as a forward-thinking restoration strategy. So on these rivers, as a lot of rivers, easy wins have generally already been achieved. Areas where landowners are keen for habitat restoration to take place, or barriers that have been removed that people were keen to get out. Now we're getting into the harder stuff, the nitty-gritty that is likely that there's some blockers to delivery of those projects. So we're really considering what has prevented delivery of those historic projects and what are the blockers to delivery and how do we remove them. So is it funding? Is it stakeholder engagement? Is it ownership of assets within the river? Is it how business models operate? Or is it just general awareness? Which of these themes do we need to look at to ensure that work can be delivered for the restoration strategy? And an example of how we're doing this is we've already funded GWCT to work within the catchment to set up farmer clusters and basically get together groups of farmers, inform them of the issues in the rivers and how things and interventions that they could do could genuinely help protect salmon and restore salmon populations. So a focus on nutrients and siltation will be obviously a big theme on that. So already building this network of landowners who are ready to deliver catchment scale interventions. And finally, I'll just summarize things. I've got to the end of my time here. It was a bit of a whistle-stop tour through the project. I could have talked about chalk stream salmon a lot more. And I'm going to circulate some resources for everybody to go into a bit more detail on chalk stream salmon and issues around them and some of the papers that I've talked about. But to summarize, Project White Hart itself is currently in its infancy, but we're hoping that the work that we're doing to remove some of these blockers that I've just talked about and being embedded locally within the catchment, backed nationally, we're hoping that it'll establish the project with local longevity that will mean we can ramp up effective restoration to benefit salmon populations after completing the catchment audit. And it hopefully means that vessels for delivery at catchment scale are in place and local buy-in and support is genuine. So fingers crossed, maybe this time next year, I can come back and reel off a list of findings and actions that are in place to hopefully save Atlantic chalk stream salmon. Right, I think that's me done.
Curt, that was fabulous. Thanks so much for that run through of all the work that you're doing. And I love those graphics too. Really make a great impact. I've seen them on your website, which is really a really good resource as well. So it'd be great if you can share the links too. So
absolutely, will do.
If you have any questions for Curt, don't forget you can put them in the Q&A and we can share all the things that he's mentioned as well. So you can go and find out more about chalk stream salmon. It's a fascinating topic. Thanks, Curt. So we're going to move on to Debbie, who's from Nature Safe. and she's going to talk about cryo-biobanking, I believe it is. So Debbie, if you want to share slides, I'll hand over to you.
Thank you very much. Let me just, oh, it's always the way, isn't it? It works fine when you've done it in practice. Oh God.
You got it.
Why?
The share button's in the mid, should be in the middle at the bottom.
I know, I haven't quite got the right.
No, this looks promising.
No, I haven't got the right. There we go.
Almost there.
Coming. There we go.
Modem with there. Fabulous.
Thank you, everybody. It's lovely to be here. Really honoured to be here to talk to you today. A slightly different chat. There will be some river connections. I'm going to be talking about crayfish later on in the presentation. But I'm going to start with taking you through Nature's Safe, who we are and what we're doing. Some of you may be familiar, but I think it might be a bit of a new discovery for some of you too. So Nature's Safe, we are a charity, conservation charity, on a mission to save animals from extinction by indefinitely preserving cells and reproductive material. And we do that by keeping them in a living state, storing them in liquid nitrogen, which is why it's called cryo-biobanking or cryopreservation. So we work with across taxa, so I know that bush dogs are not probably what you're working with on the daily, what you guys are doing, but we work across all taxa, as I say, to preserve somatic cells, skin cells, and tissues from internal organs, but also reproductive samples as well, so sperm and gametes. You probably have all heard about the Millennium Seed Bank. We are aiming to be the equivalent for endangered or threatened species, and there isn't anybody else within the UK that's doing this work. The oldest cryobank in the world is the San Diego Frozen Zoo. They've been preserving tissues for the last 40 years. So they really started it. Hasn't really developed across the world since then, but we have set one up in the UK. We're based in Shropshire. So what are we doing? What is biobanking? What is cryopreservation? So there are a number of biobanks around the world. So tissues from animals have been preserved in ultra low temperatures at -80 in a freezer for decades. People have been preserving those samples. And so what happens then is you can take blood or serum or hair, freeze it in -80, and that will enable you then to analyze the DNA. So it's fantastic research repository. The cell itself is non-functional. So it's like if you put a bottle of wine in the freezer and forget about it, the water within that bottle will freeze and expand and it will crack the glass. And that's exactly what happens when you put a cell of any kind into ultra low temperatures at -80 in this instance. The water within the cell will freeze and expand and burst the cell membrane. So the cell itself isn't functional for any regenerative purpose, but you can look at the DNA. The DNA retains intact, so you can look at the DNA for research. Really valuable resource. And as I say, it's been happening for a number of years, and there are lots of biobanks around the world, including for people and animals. So cryopreservation, this is what we do. So this is a specialist skill where we are able to dehydrate the cell a little bit and then add in some cryoprotectants or antifreeze for the layman like myself. So put some antifreeze into that cell, which means that you limit the amount of ice crystal formation, and therefore you can maintain the viability of that cell membrane. So in essence, those cells are kept in an animated living state, they're suspended in time, and they are able to be kept alive for an indefinite period. So those cells are able to be regenerated. So this is the work that we're doing. And we take skin cells. I'll come on into a minute about the samples we take, but we take skin cells and we are able to regenerate them, replicate those cells, to sort of provide proof of concept that those cells are still living. So where possible, and as I say, yes, the San Diego Zoo have been doing this for 40 years, but it's still fairly new science. And so we're trying to collect as many samples as we can from across the body. So skin samples, but also internal organs and muscle tissue, because we never quite know what is going to be the most valuable resource for the future. We have to, we work with conservation partners around the UK, so wildlife trusts, rehabilitation places, aquariums, and zoological parks and safari parks. And anytime an animal passes away, because we're not able to do live sampling, of course, anytime an animal passes away, they send us a bit of tissue, and we preserve that for them in long-term storage. So as I say, we skin samples, eggs and sperm, and ovarian and testicular tissue. So for me, it's not just storing these samples in liquid nitrogen tank isn't just storage. It is an active repository, and we are actively involved in a number of research projects because we really want to make these samples have as much value as they can for the future, for future conservationists, and to provide us with options and possibilities for restoration, population recovery, genetic rescue for the future. So this diagram is quite a nice one to sort of visualize what we're doing. A lot of the time, we're taking samples and we don't fully know why we're taking them. We just know that we can. And so we think we have a duty of care that we should. So in 100 years' time, hopefully our generations that come behind us will look back and say, well, thank goodness they saved stuff when they could. So we're popping stuff into the tank across all species. And yes, our initial mission was threatened or endangered, but we've broadened that out. So we are taking, if you're familiar with the IUCN Red List, we're taking from least concern all the way up to critically endangered. And we have got 7 species of extinct in the wild species in the tanks as well, because those least concern may not be least concern in 10 or 20 years' time, but also the point is genetic diversity. And banking genetic diversity is what's going to ensure a population remains robust. So when a population is at least concern, their genetic diversity is at the very best. So we are banking everything. And this just gives you a snapshot. So we're popping it in the tank for storage. And there are a number of applications that might be possible with those samples over time once the science catches up for unknown future recovery applications. But as I say, if we don't bank it, we know for sure we've lost it forever. Talking about the IUCN Red List, here's a visual, and the percentages are pretty stark, not going to lie. So we all know the situation. So this is why it is a race against time to try and bank as many genetics as we can. And there are a number of underrepresented taxa for this process, invertebrates and aquatic life being a couple of them. The UK state of nature, again, I'm sure I don't need to go through this with the audience here. You'll be all very familiar with the devastating statistics of our nature within the UK. And so, again, a real urgency to bring all the tools that we can to conservation and to these species preservation. So just quickly, what happens as a population gets smaller and a bit more fragmented, the genetic diversity gets smaller, species can then enter the extinction vortex, which is really, really hard, if not impossible, for a species to climb out of on their own. As genetics get closer linked, then the opportunities for stillbirths and genetic abnormalities increase, and so populations become not robust and not viable. And if any of you have heard of the last two northern white rhinos, that's exactly what's happened in that instance. So they're functionally extinct. So we've still got two on the planet, so they're not extinct, but they are functionally extinct because they are two females who are related and both are infertile. And that is the situation that we are trying to avoid. So why are we saving skin? Well, this is a question that we get quite a lot. So this may blow your mind this morning, but it always blows mine. So anyway, we take skin samples because the clever scientists have been able to do this for a few species. All cells in the body start as stem cells, and you can reprogram a skin cell to be taken back to a stem cell and then direct it to become a sperm or an egg. So then you can have a reproductive sample from a skin cell. So that for us is amazing. And the technology is there for some species, definitely not there for a lot. But again, we're banking them now so that there is an opportunity and a possibility in the future for this technology to be used. Just to demonstrate, rhino, we froze some fibroblast cells from a rhino in 2021, and then a few years later we got them out of the liquid nitrogen, thawed them and grew them up and replicated them, and you can see the replication of those cells on the second slide. So now we've got an additional resource banked for that genetic line. Also, there's cloning. Now I know the chatter around cloning, but again, useful tool, and it has been proven to be useful in conservation as well. The black-footed ferret is a good case study of this. Again, San Diego Frozen Zoo. So skin samples from a founder individual of an extinct species of black-footed ferret was preserved 30 years ago, then created a clone in 2020, and she then went on to have two very healthy pups in 2024. So they've done the same with the Swarovski's horse. 40 years ago, populations were dwindling, becoming really genetically unviable. They preserved skin cells from a Swarovski's horse, and in 2020 they cloned, and they managed to reintroduce now those old genetic lines back into the current population. And they've also done it, they've repeated that as well. So just a bit of a whistle-stop tour into some of the uses. We also are able to extract semen of post castration. So we can, if an animal either has died or is being castrated, we can extract the sperm and freeze that. And we're working with Nottingham Trent University to take a look at sperm cells, because quite often we don't know if it's normal or abnormal or what it should look like. So we're building the sperm atlas to try and increase our knowledge base about what we're actually looking at. And this is a mountain chicken frog sperm cell, for anybody that wants to know that. And these guys are really, really critically endangered, and we've got a few of them in the tank. Just again, give you a bit of a case study about what we have done and how this technology can work. So we have been freezing coral sperm with the Horniman Museum in London, and amazing project. They are one of only 4 sites that have spawning coral. And so freezing coral sperm, we are losing corals faster than we can restore them. They spawn once a year, which means that your window for regeneration and breeding is incredibly narrow. So by being able to preserve the sperm, they are able to then breed throughout the year. So we were successful. You can show the embryos on the left and then our baby cryocorals on the right. So now getting to the crayfish. So we have joined a consortium of partners to enter into some research to understand more about our white-clawed crayfish and how cryopreservation may be able to support the species preservation. As I said before, we need all the tools in the toolkit. So why do crayfish need our support? They're the UK's only native crayfish. And we are estimated to hold about a quarter of the entire global population of white claws. They were introduced in the 70s by the UK government because they thought they would be a really lucrative form of food production and coming in to be farmed. Sadly, there was lots of escapes because they're large and aggressive and very good at walking across land. And so they managed to escape from these secure sites, and then they were transported by people as well into different regions, into lakes for sort of recreational purposes. So they have absolutely taken over our chalk streams. As you all know, they carry the plague. It's a lethal fungal disease called crayfish plague. They are carriers, but it doesn't affect them or their health. However, our white-clawed crayfish are incredibly susceptible to plague, and it is deadly and fatal for our white-clawed crayfish. So this is just, again, some visual representation of how things are looking. So white-claw distribution with the red and signal crayfish distribution, and you can just see Ireland remaining signal crayfish free at the moment, so let's hope that continues. But they have proliferated across the country. There are a number of people doing amazing work with hatcheries and arc sites, so creating biosecure ponds and areas to translocate crayfish into to protect them, and create some breeding programs within those sites as well. And some good news, because it's always good to have both. So the River Allen in mid-25 found a surviving population of white-clawed crayfish in the upper catchment, despite having the signal crayfish in the lower stretches of the same river. So this is good news. There's also a big push now to do some eDNA testing and analysis, and Northern Ireland have published a landmark study late last year. They mapped 72 rivers and 38 lakes, and population is highly vulnerable, but not experiencing severe regional collapses. So this is a eDNA. They're measuring the DNA from shedded DNA in the water, which means that it's a non-invasive approach to assessing what populations are present. And more than 14,000 white-clawed crayfish have been successfully translocated or released into secure conservation sites. So the ARC sites I was talking about across various programs in England and Wales. So there are good news happening. Now, just a little bit about the cryopreservation side of things. So we've teamed up with Jen Nightingale, who I'm sure you're all familiar with from Bristol Zoo, she has a crayfish hatchery in her garden, which is excellent. And we have teamed up with University College London and the Royal Veterinary College to trial cryopreservation techniques for crayfish eggs and sperm. Now, we have done a couple of very early stage trials. It is an absolute new frontier because nobody has done anything like this before. We are having to really, it's complex, and the bigger the particle is that you're trying to freeze, the harder it is. So embryos are very challenging typically, and they release a spermatophore, which is not like a normal sperm cell. So that's a whole new area for us to explore as well. But I've got a little bit of funding through Nature's Safe to start these cryopreservation trials. And as I say, we have done a couple of sessions with that. We are also put a bid into Natural England to assess genetic diversity of natural and arc site populations, and to investigate crayfish plague resistance, because there are some instances where white claws are demonstrating resistance to plague. So we're working on those aspects as well. Just to show you, so we worked at the reservoir near Bristol. Jen got a license from Natural England so that we could do these trials on the signals as a model for the white claws. Obviously, white claws are protected, so we're using the signal crayfish as a model species for the trials. Sometimes science is incredibly rustic, so that's us in Jen's living room setting up, just it really was exploratory stage to really try and understand what we're working with. And then in the field, just looking at some embryos there. So here we are, and this is just a microscope picture of the embryos that when they got back to the lab. Nothing really to explain here, just that this is what we're doing. We're trying to understand what we're dealing with. We have done some electrospraying because what we're trying to work out, because embryos are very fatty and sort of very lipid heavy, very difficult to get cryoprotectant in because they normally have a very robust cell membrane, so very difficult to penetrate that with cryoprotectant. So what we have done, we've been looking at electrospraying to try and charge them and be able to coat them in some cryoprotectant. We have electrosprayed them. They have survived the electrospraying, and so the next phase is to try and pop them in some liquid nitrogen. Just to give you a bit of an example, because it feels very distant and different, but the research projects are going to complement each other. So we've got a PhD project going at ARU Writtle with Jimmy's farm to look at being able to cryopreserve swallowtail butterfly eggs. And so by being able, if we can crack the code on egg and embryo cryopreservation for invertebrates, then it will transfer that technology across to different species. So that was just why I put that in there. And this is our cryo store. So a number of liquid nitrogen tanks in a bit of a concrete bunker. And we have preserved 421 different species. We were started in 2020. And as I say, so the cryovials are the skin samples sit in the cryovials, and the straws are the sperm. This year we've been nominated for the Earthshot Prize, so fingers crossed for that. And that is me. Nice cheesy quote at the end there. The best time to save a species was yesterday. The next best time is right now. And my contact details are there and our website if anyone's interested. Thank you very much.
Wow, Debbie, that was fascinating. And somebody in the chat said their mind has been blown. It might not be the only one. Really, really interesting to hear, and then to see how you're using that for the crayfish has been really fascinating. Thank you so much.
My pleasure.
I think there's a question in there for you about northern white rhinos, which you can take a look at. Any more questions? Yeah, bung them in the Q&A. I'm sure there will be questions after all of that. So thanks, Debbie.
Thank you.
We're going to move on to something else now, which is another thing that I don't know an awful lot about, but I'm very much looking forward to hearing about from Sarah Teasel up in Lincolnshire. Now, Sarah, are you going to run through the slide yourself, or would you like Jack to do them for you?
Sorry, can you see me on the video? It's not
I can see your face.
Can you? Yeah, thank you. Yeah, if Jack can please present, I've had trouble.
Yeah, not a problem. I'll get that set up for you now.
Thank you.
Sarah's been having tech problems, which I think we can all sympathise with. So, Jack.
Oh, let's get rid of those.
There we go. Jack's well trained in this. So I think the combined team of Sarah and Jack, you're ready to go. So over to you.
I'll just tell you when to click, Jack, if that's okay.
Yeah, not a problem.
Hi everyone. So we've been working on the crystal moss animal, or Lephobos crystallinus project, trying to recover it since 2023. And the funding was awarded through Natural England's Species Recovery Programme. And the initial project was meant to translocate the species across Greater Lincolnshire below Wales, which I'll go on to talk about. If you could click forward, please, Jack. So first of all, what is a crystal moss animal? Because I'm sure many of you have never heard of it. So it's one of the first bryozoans ever described by Pallas in 1768. And it's one of 11 freshwater bryozoans in the UK. And these are colonial animals, and they're made up of what are called individual, zooids. And within the colony, there'll be up to 50 individuals, and the colony can get up to sizes of 50 millimeters. So as you can appreciate, they're very small. And within these colonies, they're kind of translucent and gelatinous, and they attach themselves to substrates. And these substrates can be woody debris, rocks, roots of plants like Glyceria maxima, or reed sweetgrass, and even plastic. So they're moving with the times. So each zooid has a horseshoe-shaped lophophore, which are a crown of extendable tentacles, and they help to create their own water current and bring in particles from the water column. And they're also found in lotic and lentic waters. So you can see on the picture on the right of the lophophores and the colony. And then they reproduce sexually and asexually. However, they're mainly thought to reproduce asexually. So, as I said, within a colony, you'll get so many individuals, but it'll start with an ancestral zooid, they're called, and then they'll kind of bud off, reproducing themselves. So genetically, they're the same. So as a result of this, they seem to exhibit metapopulation dynamics. So when one sister zooid dies off, potentially the whole colony can die off. And that's not just that one particular colony. If there's multiple colonies within an area, it can cause all of them to die off as well. So as a way to kind of survive this, they also produce these resistant propagules that survive through extreme climatic conditions, and then they'll hatch when it's favourable. And again, you can kind of see that on the pictures. Sorry, I can't point. So if you can click forward, please, Jack. So why did they need conserving? So there's been less than 25 observations recorded in the UK in the last 20 years, with no official records since 2012. So there's about 87 records in total, and one of the most significant is Faring's National Nature Reserve, based upon the Humber Estuary. And if you look on the bottom left map, it's kind of the top site. And this particular site has been classed as one of the strongholds, or one of the only strongholds in the country, dating from about year 2000 to about 2012. They were last seen there. And there was thousands of colonies in that area, in that particular blow well. And blow wells, just to let people know, are kind of artesian springs that are found, we think, only in Lincolnshire. So yeah, it's quite a unique site. They're believed to have suffered huge declines. So they used to be, there's anecdotal evidence suggests that they were used to be found by the pailful in places like the Norfolk Broads. And we don't really understand the drivers of decline, but we think it's the usual suspects, things like habitat loss. Like I said about the metapopulation dynamics, they do have a cryptic lifestyle. They're very difficult to see. As you can appreciate, they're very small. But because they have these die-offs, you can go and monitor them, and they'll be there 1 week, and then you go again a couple of weeks' time and they've gone. And they might not reappear for a good while. So that does make them difficult, and there's a lot that we still don't understand about them, and there's no current ornithological assessment on them. And as I said, you've got these two pictures of maps at the bottom. So on the right, it's taken, on the left, sorry, it's taken from the NBN Atlas. And this is all the historic sites where they've been found. But then I've just put this other map in from a paper from 2012 where they did core sampling, looking for the statablasts. And as I said, they're residual, they're chitinous shells, so they will survive in the core samples. But it kind of showed that actually they were found in new areas that we didn't know they were in previously. So it kind of suggests that they might just be chronically understudied, and there's no current population assessment for them, unfortunately. So that was kind of what we were dealing with going into the project. So if you can click forward, please, Jack. So we started by surveying 10 blow well sites. So the historic stronghold at Faring's National Nature Reserve, which is pictured in the bottom left, and then 9 receptor sites that we thought could be potential potentially good sites. So we combined habitat suitability assessments along with presence-absence surveys. And at the time, I'd contacted Nottingham University Professor Dr. Hartikainen, who's a specialist in bryozoans, and she happened to be developing a species-specific eDNA for Lithopus. So it was a really lovely timing, and it was super helpful. They really wanted to come on board with the project, so that was amazing. So we also did eDNA testing at each of the 10 sites. We were also approached at the start of the project by The Deep, who are an aquatic conservation charity based in Hull, and they conducted seasonal water quality monitoring across 10 sites over the course of a year for us. So again, that was amazing. So the outcomes from that were, can you please click, Jack? So unfortunately, we found no physical signs of any colonies at all 10 sites, which particularly for the Faring site was, it put a spanner in the works, so to speak, for the project, as you know, everything hinged on finding this population that we would then potentially translocate. We did, however, get one positive eDNA sample from Farings. However, we had to remember that this was a new assay, so we weren't sure of its efficacy at that point. However, overall, from all the assessments that we did, it kind of came to light that there wasn't really, within the 9 receptor sites, there wasn't many that were going to potentially be suitable anyway. So we kind of had to go back to the drawing board with the project. So if you can click forward, please, Jack. So because we'd formed this amazing partnership with Nottingham University, we were able to continue on with the eDNA testing. So we started surveying nationally, and we looked at both historic sites as well as favourable habitat sites. And the favourable habitat sites were kind of based off another paper that had looked at some statistical stuff that we couldn't replicate, but things like elevation, land use, tree cover, things like that. So we kind of were shooting in the dark a little bit. But we roughly surveyed 60 sites in total. And we also continued walkover surveys and sediment sampling with, we set up a volunteer scheme to do continuous monitoring of the below wells. That was hopefully, at some point we might find them. We also did debris sampling and walkover surveys at historic sites. And Jack, if you can click forward, please. Oh, back. Yep. Oh, back again. Sorry, just once. Yeah, thank you. Sorry. So the outcome, there was, we found positive detections at 12 locations across 8 counties. So you can click now, please, Jack. Thank you. Yeah, so you can see on this map here, the green is where we had positive results, the red negatives. So as I said, these were at 12 new locations. So you can click off it. Now, Jack, was just to give you an idea of where they were found. But we also got a continued signal at Farings, so suggesting they were definitely there. And we also had another breakthrough of 30 start blasts found on the River Foss and one found on Burton Mill Pond through debris samples. And that's then pictured in the picture below. And then right at the end of the project, we also managed to get some passive debris traps to try and capture some statoblasts at Farings. And the bottom right picture is The Deep, who'd fashioned this egg collector that they use for corals. So that was cool. So it was a really good outcome in the end for what started as a bit of a disaster. We changed our tact and we got some really good results. So if you can click forward, please, Jack. So as a result of this, we then applied for further funding through Natural England Species Recovery Programme again, and we were successful. And in June 2025, we decided one of our first steps would be to revisit some of these positive sample sites. And we'd got quite a high signal from the Belton Estate, which is based in Grantham in Lincolnshire, and it was quite close to us, so it was easier to access. So we all met up on a very rainy, soggy day, and you can see it in the bottom left corner. And we thought there was absolutely no chance we were going to find any. And Dr. Hartikainen came with us, and she managed to find thousands of colonies. So it was absolutely incredible. It was one of the biggest wild population finds in over 20 years. And if you can just click, please, Jack. So this is the outlet that leads into the River Witham, and it's called Torthorpe Hollows Pond. And you can see on the picture on the bottom panel, the second picture in, this is the colonies that you can't quite make out on the pond picture, but they were just covered over the entire pond. It was like a nursery for them. It was absolutely incredible. And so from that, we then transferred colonies to the University of Nottingham to start some ex situ trials to hopefully go towards breeding them. And then we went back at a later date and we carried out some systematic surveys and substrate trials. If you can click, please, Jack. Oh, click again, sorry. Yeah, so this is the pond they were found in. So where it says colonies, this was kind of the little outlet they were in. And we sectioned up the pond, and we had some preliminary sample, eDNA sample, to suggest that they were further back in the pond. So we wanted to look right across it, doing walkover surveys, eDNA sampling. We also did some water quality testing and kind of habitat assessment to look at the different habitats that they might, like niche habitats they might be inhabiting. And from this image, you can see that there should be another pink dot under the upper sample, but the blue dots are where we got positive eDNA samples, and the pink dots is where we did the walkover surveys, and unfortunately we didn't find any. So again, it kind of exemplifies the fact that they're so difficult to find. However, we did learn that within them areas, it did show that it was kind of suitable habitat still, so that was promising. And if you can click forward, please, Jack. So, and again, sorry. Yeah, so we also did the substrate trials where we used proxy substrates. So I chose metal, wood, plastic, rope, and stone. And within them, we placed them in the outlet, and we wanted to see how well they settled and colonized them. And if you can click forward, Jack, please. And again, sorry, there's always a space. Yeah, we identified that natural rope was the most, oh, sorry, I've lost my thing, was the most productive material, and that they attached readily and survived. And they had the most colonies on them compared to all the other substrates. Like they all colonized, but when we moved them into other areas of the pond, the others died back where the rope didn't either. So this gave us a really good signal for future translocation. And also it was a real breakthrough because it provided substantial living material for ex situ breeding. And we also went on to do genome sequencing, which was part of this next project. So if you can click forward, please, Jack. So as I said, the University of Nottingham then were carrying out genome sequencing from the Belton wild population. And what they found was that the genome displayed extremely high homozygosity, which basically means really low diversity. So it was kind of explained to me, it was like a brother and a sister mating and producing offspring. So it suggested, yeah, high inbreeding or potential population bottlenecks or founder effects. They also looked at the COI mitochondrial gene and sequenced that across 4 locations, plus 2 European locations of Italy and Switzerland. And again, they found 0 variation, which was really unexpected. And it suggested either highly conserved lineage spanning across European populations, or that perhaps it wasn't a reliable marker, which it often is. So it meant that the University of Nottingham kind of had to go back to the drawing board, and they came up with a 20 marker microsatellite panel, which allowed for future genotyping of nuclear diversity. So the tool now exists for us in the next phases to hopefully continue to look at that genetic diversity, which is obviously going to be super important when they do have such low diversity in terms of translocation and things like that. So if you can click forward, please, Jack. So we've, like I said, we had a volunteering citizen science program from the beginning, and in this part of the second project, we did a really good volunteer engagement day where we built statoblasts that were taken from a paper, and they were like kind of plankton nets that worked really well, and we deployed them at Farings. And you can see in the bottom left corner one of our amazing volunteers and proudly holding up the trap. And after 2.5 years of searching, volunteers and the team recovered 2 Lophopa statoblasts, which doesn't sound like a lot, but it was like crucial physical evidence that we needed of continued species presence, and it was just amazing after so long of looking for them. I also surveyed along the River Derwent and the River Delf, and that yielded a further 11 stratablast. And again, these can be used for further genomic work as well as potential hatching. So that's amazing. And then we've also continued doing monthly eDNA sampling at Farings, which is, again, providing a temporal data set for tracking the population persistence as well as meta-population dynamics. And then if you can click forward, please, Jack. Thank you. So we also did habitat suitability modelling. So the MRES matched occurrence data with environmental parameters across a broad set of sites, and basically using bioclim data, so bioclimatic data, and we had about 8 variables, one being elevation, and then other things like highest precipitation per month, I think, or wettest. Anyway, I've forgotten what it was called, but it showed us that the species doesn't like large amounts of rainfall, and that it was less likely to occur in areas with high seasonal temperature swings. And it was the first model done for the species in the UK. And you can see on this map on the right-hand side, the darker pink basically shows where it's more suitable. Now, this is a preliminary map, and we hope to take forward this map and use more parameters and further like habitats that we can then refine the model further and further. But it also showed that it directly identified Belton Estate as a highly probable site, which was consistent with the 2025 discovery. So it was helping to validate the approach. We also identified a gap. So the eDNA presence absence data is the most valuable input. So since the beginning of this second project, we've been in conversations with the Natural England and the Environment Agency, and we're hoping that we can eventually get the eDNA assay incorporated into their monitoring programs, which would be an amazing national effort. If you can please go forward, Jack. So then finally, as part of this project, we've managed to establish a nice ex situ breeding programme. So the University of Nottingham, it was kind of touch and go to start with, so they're quite a difficult species. And at first, when we took the wild colonies ex situ, we found that they were kind of dying off. But now Dr. Hartakainen pictured here in the left, she's managed to find that Cryptomonas, which is a microscopic algae, is a good food source for them. So we also went and visited the deep. So this is us at the deep, and we brought a colony over and we brought some Cryptomonas for them, and they're now establishing a cultivation of Cryptomonas because it's quite slow growing and you can only buy it in small amounts and it's expensive. So they're establishing this phase that they can then lead on to breeding at the deep as well, which is again going to be a really important aspect of the next phase. And in the bottom right corner, you can kind of see they've just set up. This is newly been set up for hopefully breeding of Lithopus. And if you can just click, please. Yeah. So this is. I just wanted to include this. This is a really beautiful picture we got under the microscope of Lithopus, and yeah, I just think it's really lovely. And then if you can click forward, Jack. So and then here's a little video of him. If you just press play. So I just wanted to show you this because it gives you a really good example of them. So you can see the kind of lophophores, and this is where we disturbed them, and you know, they've retracted them and then they're bringing them back out, and it just looks really beautiful. And you can kind of click forward, Jack, towards the end. We won't watch it all. So you can see them here, like using their individual tentacles to pull in their food sources. And then the kind of orange, yellowy orange bits are their guts. So I just thought that was a cool video to show you guys. And then if you can keep clicking forward, Jack, until it goes to the next slide. And then finally, I just wanted to thank our partners, which without them, we wouldn't have been able to do the project, especially Nottingham University in the Deep. They've been amazing. And just to say, we were finalists for the BIASA Great British Wildlife Restoration Awards this year, which was amazing. And it reflects the national significance of what we've achieved in the project. And we have got another project, hopefully going to happen soon, but it's not finalised yet, so I can't say too much about it, but hopefully we'll come back at some point maybe and tell you more. But yeah, thank you.
Wow. I'm having a real learning day today. I've never had so many presentations talking about DNA and genomes. Yeah, mind is still blown. Fabulous, Sarah. Thank you. And those last videos were just mesmerising. Fabulous to see. Thank you so much.
Thank you.
So for our final presentation, we move on. We've looked at sort of species from the small to the relatively big. And Sarah Perry's going to talk us through some work they've been doing looking at protecting the habitat that's needed for all the species that live in chalk streams. So Sarah, over to you.
Thanks, Alison. Hi, everybody. I'm just going to share my slides. Go. Right. Hopefully everyone can see my presentation okay.
Yeah, that's come up. It's not in presenter mode yet.
No, it's getting there. There we go. Is that all good? Excellent. Okay. Thanks very much. Okay, well, hello everybody. Hopefully can keep you all interested for the last 15-20 minutes or so. I'm Sarah. I'm the River Recovery Manager at the Herts and Middlesex Wildlife Trust, and I oversee our programme for restoring chalk streams across our Hertfordshire landscape. And I have a wonderful team who do all sorts of different interesting things relating to catchment support and citizen science, which you may hear about another day. So really, coming in at the end, this is really to frame some of our work that we've been delivering over the past, 14 years really, but with a real focus on restoration efforts over the last 8 years since I've been in post, which is really providing the groundwork and the base habitat condition to support all of these wonderful species that we've heard so much about today. And to bring it home to a Hertfordshire context, I'm sure there's lots of chalk stream buffs obviously in the room, but in a Hertfordshire context, we have 3 main chalk stream catchments. We have the Colne catchment out to the west, which is filtered into through the Chilterns chalk streams, some of which fall in Hertfordshire, some of which fall over the border into Bucks and Oxfordshire. We have to the east our main chalk stream catchment, so this is the River Lee catchment, which entirely falls within Hertfordshire, and which is where later on in my talk I'm going to be focusing on exemplifying some of the work we've been doing in those chalk streams over the east side. And then we have a couple of rivers in the north of Hertfordshire flowing northbound into the Great Ouse catchment and out ultimately into the Wash out in Norfolk. So lots of different river systems. These make up around 600 kilometres of chalk-derived waterways, whether that's main river, feeder streams or smaller tributaries, as well as some of our headwater windborne sections as well. And of course, we've heard some of the wonderful wildlife that our chalk streams nationally support. And in a Hertfordshire local context, we have been working through the local nature recovery strategy to pick out the target species which we really feel in a Hertfordshire context are the key to restoration success. So we've picked out a suite of 8 species across birds, mammals and invertebrates, and we really are using these as a beacon, a flag, to engage landholders, engage communities with the reasons why we want to restore our chalk streams. You know, we can't necessarily just think about these species in isolation. Obviously, there's lots of projects seeking to reintroduce them, and that's fantastic, fantastic work. At the same time, it's really important to get the habitat conditions right that will lead to long-term resilience for these key species. So probably notably, we don't have a version of chalk stream Atlantic salmon, which Curt talked about earlier in our Hertfordshire catchments, but we do have critically endangered European eel, which are of course IUCN and red listed, and we've seen 75% drop off in our Lee catchment alone. And Alison's going to talk a little bit later about another partnership project that we and others are part of to try and bring the European eel back across the Thames River Basin. And our rivers, where they do exist, are not in good condition. I'm sure this is a familiar story across many of your chalk streams as well. Really sad story. Our rivers have been incredibly impacted over the last 200, 300 years by us and the activities that we've put upon them. So we often now see channels that are degraded, they're lacking their natural gradients, they're over deep because they've been dredged, which both degrades the channel and also disconnects the river from its wider floodplain. We're plagued with invasive species in Hertfordshire as well. So signal crayfish, yes, absolutely. They are ubiquitous across our river catchment. And in fact, our last record of white-clawed crayfish in the eastern part of the county, in the Lee catchment, was back in 1996. So we know there are huge issues with invasives that are exacerbating some of these other pressures. We end up with really homogeneous channels that don't have the ability to move and deposit sediment in a natural way. So we've got species that are just about surviving, but are locked into these really degraded, long linear channel sections that we really need to do something about if we are to secure them for the future. And a couple of years back in 2020, in line with the National State of Nature report, we pulled together our own State of Nature report for Hertfordshire that looked at wildlife trends over the last 50 years between 1970 and 2020. And this was really to understand what trends we were seeing in terms of wildlife declines and losses, as well as trends in wildlife moving into our area as a result of climate change. We also considered how land extents had changed across that time period. And I think some of these figures are really stark. So if we take our core habitats in Hertfordshire, they only represent 16% of our whole county area. And of that 16%, just 5% is wetland, which is far less than the national average, about 2/3 less than the national average of 12% cover. So we know that wetlands are in really short supply, and those wetlands include our chalk streams, riparian wet woodland, ponds, reed bed and feeder streams, as well as wet grassland. And linked to this, we can see that 24% of our species of conservation concern in Hertfordshire are linked to wetlands. So about a quarter of our species of conservation concern are dependent on wetlands to thrive. So without doing something to restore the quality and condition of these wetland habitats, as well as extend their coverage, aiming for sort of around that 24% mark, we know these species are not going to really recover and become resilient in the face of ongoing pressures through things like climate change. So this is really where our role comes in, and our Wilder Rivers programme, first branded Living Rivers, has been running since 2012, which was in the first stage is really about raising awareness of chalk streams. You know, all the good work that everybody on this call and Alison and CaBA and the Rivers Trust have been doing over that time to really raise the flag for chalk streams has really helped with gaining public consciousness about these rivers. It was very uncommon back then to actually go out and speak to people and have any sort of sense that people knew what a chalk river was, to be honest. Whereas now, most conversations I have on the riverbank, people will say to me, "Oh, did you know this was a chalk stream?" So it's really great to hear that. And as a result of that, our work has really moved on from that education, campaigning, awareness raising aspect into doing all of that, but also delivering a lot of this priority restoration work and really supporting landholders to think about how these rivers can be restored in the different contexts that they sit. There's no one stretch of river that's the same, we all know that, and indeed there's no one landholder that's the same. So every project that is developed has to be considered in that wider context. In the last 2 years, so in 2024, we were really excited to receive a biggest chunk of money that we'd had sort of injected into Hertfordshire for river restoration in the past, a £1.7 million DEFRA Species Survival Fund grant, and coupled with around £600,000 of local match funding through a blend of sources like Environment Agency, Highways England, local landholders as well, we were able to put together or start to deliver a long-standing programme of work on 6 of our chalk streams. So including the River Ash, the River Lee, Mimram, Quin, His and Stort. And this consisted of around 8 different capital projects to restore the rivers, around 11.5 kilometres in total, as well as look to the wider floodplain, reconnecting the river to that floodplain, creating wetland habitat and features within it. So the river has that long-term resilience, and we were expanding that wetland coverage towards that 24% that is needed to support our target species. Doing a bit of a deep dive as well into the data, we found that around 109 freshwater species were actually dependent on these rivers in these project areas. So yes, we have our LNRS priority species that are our beacon, and if we get the rivers right, we should see those species thrive. At the same time, we hope that will have a trickle-down effect to a number of these other 109 species that sit below. Clearly, from the talks we've heard today, some species will need more intensive interventions, and that has been picked up through the LNRS as well. Thinking about species that won't necessarily come back just through restoration work alone, that needed targeted action to help them. And we're working with the LNRS partnership to consider that and how we deliver that going forward. And so the next couple of slides really are just going to pick out one of those suite of 8 projects to show you some of the types of restoration work that we've delivered across that wider programme in the last 2 years. And picking on the River Ash, which is one of our chalk streams right to the far eastern side of our county, which really is, it's a chalk stream, but it has a lot of characteristics that are defined by the overlying clay that we find in quite great swathes in that catchment. So it's a much flashier chalk stream than some of our western counterparts, which means the response of the river to river restoration can be quite different actually to some of our more typical bucolic chalk streams. And so thinking about that as part of our restoration planning journey and of course delivery and adapting our delivery to suit those conditions was really critical to this project. And I'm really, really pleased to say that after a lot of hard work by loads of different people, including the landholders for certain, our funders, contractors who I've worked with, the team, and volunteers, including monitors and practical habitat volunteers. Massive joint effort. We were really excited to be awarded the River Restoration Centre UK River Prize this year, which again is just a nice nod to chalk streams, raising the awareness of the wider rivers community and putting a marker down for how they might be restored across the board. So just a few pretty pictures really now to walk you through. So all of the work was really driven by process-based restoration. So we were not in intention trying to create isolated habitat features. It was thinking about how do we address the underlying pressures that are impacting the river? How can we overcome those pressures to unlock long-term resilience and habitat stability, so both the species that depend on these rivers can thrive, but also so that the rivers and their catchments are more resilient to environmental shock incidents, such as the heat wave we've just had last week and penciled for end of this week, as well as some of the massive winter flooding that we now see in our chalk stream catchments, especially the ash with the overlying clay. So it's really about that process-driven restoration, trying to, wherever possible, think about large contiguous areas and really moving towards 3 key principles that drive that restoration work. So firstly is unlocking and restoring bed gradient. So this is looking to address the impacts of dredging and of barrier creation, things like fords, weirs and culverts on our river systems. Considering how we reinstate natural planform, so where rivers have been straightened or realigned and moved off the floodplain bottom or even canalised, how can we undo that straightening pressure? How can we reconnect the river to its wider floodplain, but also the groundwater sources that it may have moved off of when it was realigned? And lastly, floodplain reconnection, so enabling and encouraging seasonal inundation into the floodplain, so it can both hold, the landscape can hold more water at times of higher flow and more quantity, and also then act to recharge the river as well as the aquifer in the leaner times of the year. And so this aerial photo looking upstream, I wonder if I can use my little arrow guy. No, I'll have to describe. So this is a photo taken of the upper River Ash. This is at the very start of our 7-kilometre reach on this chalk stream that we restored with the Easney estate over the last 3 years. You can see a sort of new-ish wiggly channel towards the middle left of the page. This was the original paleo channel, so the original course of the river that had been filled in at a point that a railway was built, which ran historically along the tree line to the left of the page. The river was straightened into a channel which sits sort of perpendicular to the paleo channel and realigned off of its valley floor bottom. As you can imagine, that chalk stream channel looked very much like that first picture I showed you. Deep in size, sluggish, silty, full of burry, none of your typical chalk stream plants. But what was quite interesting was between the planning stage and sort of working these designs up with the landholder and delivery stage a few years later, water vole, one of our key species, had actually found its way back to this upper part or middle upper part of the catchment and was doing pretty well, only in that dredged and incised horrible bit of channel. So we had to work to adapt our plans, and I picked up this paleo channel using LiDAR, which we hadn't really considered at the start of the journey, mostly because ambitions over the life scale of the project had grown. The sort of understanding of the landholders, who've been amazing and really committed and supported, really grew, and it really, I think, gained their excitement, and they opened up to doing a bit more and experimenting a little bit. So this paleo channel is a bit of an experiment. We've created it as a connected backwater for now, partly to comply with our permit conditions, also so that we don't dewater the existing straightened channel where the water voles still have a stronghold. And instead, we are enabling the river at its upstream end, sort of going in towards the middle of the screen. You see a cluster of trees where the new paleo channel reconnects. We've really lowered the bank there, and we've put in a huge gravel bar with a load of dead wood, with the view that over time the river ash will start to break through and naturally pick up the smooth gradient paleo channel course. And it was really exciting to see it doing this over winter this year. And this is a photo of just that, taken in February earlier this year. That was just 5 months on from when this work had been completed. So quite early days, quite rough and ready with the non-vegetated edges, but you can see that chalk stream starting to divert away from its straightened linear channel into this new sort of its old paleo channel. And we're quite excited just to watch how that develops over time. We've got quite a lot of monitoring and surveying going on just to track change and see if we can sort of prove concept, enabling natural process to take hold once more. The last sort of principle that our restoration is based on is floodplain reconnection. So as I said before, enabling that river to come up and out and then recede back in. And you can see it doing that exactly here. So those scrapes and ponds that we created within that floodplain mosaic, holding water in the winter months and starting to naturally draw down in the summer months, which provides great feeding grounds for our wading wetland birds like lapwing or redshank, which we do still have in isolated patches in the county. And really giving back to the river in those leaner times of year. So we still see that we're, the areas of meadow that were wetted in the winter holding much more soil moisture, even now in these higher temperature times of year. So that's probably the final principle. Where we're looking at unlocking and restoring bed gradient, unfortunately, we're not always able to do that with the materials that exist in the channel as they are now. We have to reintroduce quite a lot of gravel. Our rivers have been ravaged for gravel in Hertfordshire. There's a huge standard gravel extraction industry in our region, so it's very much about putting that gravel back. And where possible, we always source that from the floodplain, so we know that we're using the gravel natural to that catchment, whereby species within the river system may have naturally evolved to using that gravel, such as brown trout. So this is just a bit of a picture to show what that operation looks like. Huge undertaking, but what was really nice was we were able to align this with the farmer's ambition to put this whole field, it's about 3 hectares or so, into a countryside stewardship wildflower mix. So we extracted gravel, cleaned it all, threw it back in the river, and then levelled it all out. And within a month or so, they drilled it all for AB8 wildflower mix. So quite a nice sort of overlay of different intentions and how you can work together to achieve nature benefit. How am I doing on time, Alison?
Not much left.
Not much left.
No, we've only got 5 minutes on the call overall. So if you can wrap up in a couple of minutes, that would be great.
I will whiz through. That's absolutely fine. So this is just kind of an after shot to show you what some of those regravel sections look like. So you can see that our starwort, one of our native plants, is starting to recolonize these gravel riffles. And what's quite exciting is to see how quickly that process can happen. Within just a couple of months in some scenarios, the plants have started to recolonize at the edges. The water crowfoot had found its way downstream and rooted on one of the large pieces of wood we put in and started to take hold. The landholder has been brilliant. So the Buxtons, they've been helping transplant patches of crowfoot with their sons over summer, and we did undertake quite a lot of marginal planting, so of the banks that we regraded, just because this catchment is so flashy, and so we wanted to ensure there was a rootstock in those friable banks going into the first winter post each restoration phase. So it does take quite a few years for those plants to establish, which we are starting to see 2 years on, so that's quite encouraging. Unlocking fish passage. So again, using some of that gravel, we were able to create fixed riffle systems downstream of immovable structures, such as this ford, which is the main access route to both sides of the farm. Quite a historic feature. We didn't want to, we couldn't really remove it in its entirety. So instead, it was notched a number of years ago by the EA. Wasn't quite functioning, we don't think. We think due to washout downstream, the height differential really between upstream and downstream was so great that fish may not be able to move up and over. So we've instead created this fixed riffle system to raise the levels downstream, and we now have about an inch difference of water that fish are able to zip up and over, and we've seen fish doing that, so it's sort of proof of concept it is working, and that's naturalized very well and looks more like one of those riffles that I showed you just a second ago. Again, looking at paleo channels, so just another view of the paleo channel. And again, we picked up a number of other relic meanders that have been filled in over time, choosing instead to keep the main channel and provide these paleo meanders as high flow refuge areas, which I'm really pleased to say were used as part of a grayling reintroduction scheme by the Environment Agency. When they came out to the river in May 2024, the river was in full spate, would you believe, in May, and these were the only safe areas that they could reliably release the grayling fry into. So quite nice to sort of show how they can benefit wildlife in the channel. Just another pretty picture of some of the bank regrading.
Sarah, sorry.
No, I'm done.
If you could whiz through any remaining ones, just show us the pictures. But yeah, I think we've got to bring it to a close.
Backwater, lots of large wood, volunteers doing their thing. And finally, just a little bit, just to say we are monitoring these really intensively. Some really exciting early results, including river fly abundance increases up to 177%. We've seen improvements in flow, silt, and quality indexing. And some of the key species, it's been really fascinating to watch return. So trout respawning on gravel that had only been laid 3 months prior. And all of these photos, just to say, have been taken on the river ash. So that trout is a wild river ash trout. And I'm reliably informed that they are genetically distinct, and some monitoring of their scale samples went on a few years back. To show that they have speciated in Hertfordshire down to river level. So similar to what Curt was talking about with the salmon, trout clearly do the same thing. So really important to preserve those genetically distinct populations. And that's it.
I'm glad you managed to get genetics into your talk as well, Sarah. Now, I have chaired really badly, and we normally leave time for Q&A. I've just allowed our speakers to talk because I can see in the chat that everybody has found all the presentations fascinating and really interesting. We've had a few questions come in. I'm going to take those away and ask the speakers to respond to those separately because we are just about out of time. But yeah, thank you so much to all 4 of our speakers. You've really inspired and educated, I think, probably more than any of the presentations, webinars we've run. People have really sort of valued learning something new, and certainly there's been a lot of that in those talks. So a huge thank you to our presenters. I know how much time it takes to put these slides packs together, and then the nerves of running through them. So you all did an amazing job. Thank you so much. So just to bring us to a conclusion, that's the end of this webinar. If you have ideas about webinars that you'd like to see in the future, then please do drop me a line. And also, if you want to watch back any of the fabulous speakers we've had in the past, then you can go on the CaBA Learn webpage and you can see the rest of our webinars and other ones in the CaBA sort of group as well. So I'll draw that to a close by saying thank you very much. Thanks for attending as well. Really great to see so many people interested in staying right to the end. Thanks to Jack for helping us out too. Enjoy the rest of your day and hope to see you at the next webinar. Thanks very much.